Far too often, treatments and policies are developed to address cancer, not the people living with cancer. Friends knows that centering patients, their needs, and their quality of life can inform better research and change the trajectory of science. We're looking to the future of cancer care, bringing patients and their advocates to the table, making them partners in leading the charge. Our work, advocacy, and expertise is driving groundbreaking legislation that gets patients the treatments and care they urgently need faster than ever. Clinical trials historically have been restricted to a small number of patients. We're broadening eligibility criteria in cancer clinical trials to make them more accessible and equitable. Diagnostic testing can be inconsistent and variable. We've brought all players to the table to make sure tests are accurate so that personalized medicine can truly be precise. Too little information was being used from real-world patient experience to inform treatments. We're creating a new paradigm to shape how therapies are developed. And now we're charting a path for the next generation of breakthroughs. Breakthroughs like our CT Monitor project is one of the largest scale scientific endeavors in Friends history to assess the ability of a rapid and easy-to-use blood test to monitor treatment response. We're setting the standard for a new biomarker for use in immunotherapy, which will support informed decision-making for patients. We are transforming lives, and we'll use these 25 years of momentum to drive even more life-changing breakthroughs for years to come.
Good afternoon, and thank you for joining today's virtual meeting, The Next Generation of Cellular Therapies, Opportunities to Accelerate Development. Today's meeting represents the next step in a series of projects in our work to advance cell and gene therapies since 2018. We're delighted to bring together a dynamic group of panelists today to explore strategies for developing the next generation of cell and gene therapy products by leveraging recent scientific and clinical advancements to help inform evolving regulatory policy. In addition to the panelists that you'll hear from today, numerous experts have been involved in an initial working group leading to today's discussion, and we thank them for their time and expertise. During our forum today, you'll hear from some of the working group's ideas and potential opportunities as part of the panel presentations. In addition, they've begun to work on a white paper that will be distributed for our subsequent public meeting, which will take place on May 22nd. As a reminder, our presenters and members of the working group will be answering questions during the meeting today. To ask questions, please use the Q&A function at the bottom of your screen, and they will get to as many of the relevant questions as possible during the panel. Before we kick off today's discussion, I'm happy to welcome Dr. Peter Marks to provide some opening remarks to get us started. Dr. Marks is, of course, the director of the Center for Biologics Evaluation and Research, or CBER, at the FDA. He's been instrumental in this partnership since the very beginning and is a true visionary in the field. It's my pleasure to now turn things over to you, Peter.
Thanks so much, Jeff, for that kind introduction. And thanks for inviting me to talk a little bit about the cellular therapy products and our perspective on their development. So to date, there are now 6 approved CAR T-cell products in the United States. 4 of these are for non-Hodgkin's lymphoma or acute lymphoid leukemia, and 2 of them are for multiple myeloma. We've seen these products gain somewhat wider use as they've moved into, in some cases, into earlier lines of therapy, although they are still all being used in the relapsed or refractory disease setting. That said, there is so much possibility here for both autologous and allogeneic cellular therapies that it really is a very exciting time. I think we see an ever-increasing number of submissions of cellular therapy products to the agency. In fact, last year we had close to 100 submissions of CAR T-cells in various applications. The majority of these are still in hematologic malignancies, yet we're seeing an increasing number being submitted to the agency for solid tumor indications. And in terms of the solid tumor indications, the antigens are scattered over about 60 different targets or so. The number of targets that we continue to see the majority of the applications for, though, are close to the majority, are still the traditional targets in the hematologic malignancy field, including CD19 and BCMA. But that said, I think the real excitement in this field comes from the fact that with the advent of CRISPR-Cas9 genome editing, the accelerating technologies around the ability to manipulate cells to potentially have allogeneic chimeric antigen receptor T cells or other cellular therapies available for use. I think there's a lot of excitement here, in part because once one can make multiple genome edits into these cells, one can potentially have more complex constructs in an allogeneic package. And so it'll be interesting to see how the field develops over the coming years. Probably we'll continue to see a fair amount of autologous chimeric antigen receptor T cell use and development, but be interesting also to see how this flows on the allogeneic side as well. And in this regard, I think we'll look forward to working with sponsors as they move forward development programs, trying to expand out of hematologic malignancies into the solid tumor realm, and also trying to take the therapies even within hematologic malignancies into earlier lines of therapy where potentially they may help prevent relapse from occurring. So at FDA, we've been trying to help move this forward in a variety of means. We have put out a guidance on considerations for the development of chimeric antigen receptor T cell therapies. It's a draft guidance that will ultimately be updated and revised and issued as a final guidance. Additionally, we are really looking to have conversations with sponsors who are developing novel therapies. In kind of stepping back and thinking about, for us, where there's room here, I think, for some real work together to move things forward. I think as we've seen chimeric antigen receptor T cells and other cellular therapies advance, what we're seeing is that the bottlenecks sometimes in development that we see have to do with manufacturing. Although obviously there's a benefit risk consideration for manufacturers about when they pull the trigger on true commercial process manufacturing. In general, and I don't mean to overgeneralize, sometimes it just seems like that transition is happening later than it should. People may want to consider preparing for success. It's a good thing to have some dialogue over because we have seen the challenges that can occur when the transition to commercially viable processes is later than it should be in terms of delaying products coming to market. So something just to consider. Another thing I think to consider as we're thinking about these products is whether or not clinical trials could be advanced in a different manner, whether we should be starting to consider basket trials for these products, because after all, they are targeted against certain antigens that may be present on multiple tumor types. And if one could get those data in a basket trial, one might be able to have a broader label to start. And then finally, just as a word of advice or a welcoming word of advice is, please, I think, feel free to contact us and engage in dialogue about novel development programs. I think just to conclude, this is just an incredibly exciting area because having witnessed now the deployment of CAR T cells and seeing now people who have been treated with them a decade ago who appear to have been cured of their disease, in a manner that may be much less toxic than some other methods. It is an exciting place to be, especially also seeing what might be possible in other realms of cancer. So look forward to the rest of the discussion today and the other talks, and I'll turn it back over to Jeff.
Thank you, Peter, for a terrific introduction and starting point for today's forum. To begin our discussion today, we've asked two of the working group members to share experiences from two slightly different vantage points. First, to provide insights from an academic institution at the leading edge of cell therapy development, I'd like to turn it over to Julie Jadlowsky from the University of Pennsylvania. Following Julie, we'll turn to Jonathan Jazayeria from Kite, a Gilead company, to share an industry perspective, specifically regarding the experience of developing subsequent generation cell therapy products in Yescarta and Tecartus. Julie?
Hello, my name is Julie Jadlowsky. I'm the Director of Translational Science at the Center for Cellular Immunotherapies at the University of Pennsylvania. And today I'm going to give a brief talk on leveraging existing data to benefit cell therapy programs from an academic perspective. So a little bit about our CCI. We focus on first-in-human pilot and phase 1 T-cell-based cellular immunotherapies. We have a single manufacturer, which is our UPenn Clinical Cell and Vaccine Production Facility, or CVPF. Our CVPF has supported manufacturing of approximately 28 T-cell product INDs since the year 2000. We have a standardized CAR T-cell manufacturing platform. We have submitted this under a drug master file to the FDA to allow them to see our comprehensive programmatic data, and it also helps streamline IND submissions on our end. We have a standardized lentivirus production platform, so we use the same packaging plasmids across vector lots, and we also use the same vector backbone across all of our programs as well. We have several instances where we have used a single product in multiple disease indications, and our CCI safety and pharmacovigilance team reviews and monitors safety across all of our cell therapy programs. The centralized, consistent structure has allowed us to leverage programmatic data to help advance individual programs or justify reduction in regulatory burden. So today I'm going to talk a little bit about some of our successes and some of the work we have in progress in trying to leverage some of our programmatic data to make things a little bit easier for everybody. So we have leveraged programmatic product stability data to reduce or streamline our clinical testing program. We've used clinical data to justify doses, determine expected safety concerns, and develop toxicity grading and management algorithms. We've reduced or eliminated RCL testing of product and patients. We're currently working on using persistence and safety of modified cells to reduce patient follow-up duration. And it's these last two things that I'll spend my short time here talking about. So, starting with replication competent retrovirus or lentivirus, program-wide data on RCL testing of vector lots, cell products, and post-infusion patient monitoring was published by our group in Marcucci et al. In 2018, which affirmed no evidence of RCL. This and other data helped to inform the January 2020 guidance from the FDA entitled Testing of Retroviral Vector-Based Human Gene Therapy Products for Replication-Competent Retrovirus During Product Manufacture and Patient Follow-Up. And within this guidance, there's two passages that I would like to bring to your attention. First of them being, if you have accumulated manufacturing and clinical experience that demonstrates that your transduced cell product is consistently RCR negative, the agency recommends you provide this data to support reduction or elimination of testing ex vivo genetically modified cells for RCR. The second passage is, after you have accumulated patient monitoring data with your product, you may provide a rationale to discontinue all active testing of patient samples for RCR in the safety monitoring section of your clinical program. So we in CCI saw this as a door opening opportunity for us. So we decided to forge through that. And I'll share with you some of the justifications that we used for making some changes. So from a product testing perspective, per our publication, RCL was not detected in any products manufactured by CVPF. This included 17 different vector lots under 8 different INDs and 375 T-cell products tested by qPCR or biological assay. At the time of our request to the FDA, which happened around July of 2020, the number of products was up to approximately 705. Additional justification also came from the Cornetta group reporting 460 transduced T cell products across 26 clinical trials, which were RCR/RCL negative. In terms of patient monitoring, in our publication, we evaluated 308 patients, which was almost 195 years of cumulative test follow-up. We estimated that using a Poisson probability model that a single patient or group of patients would need to be followed for almost 53 years to observe one positive RCL event. This highlighted the unlikelihood of such an event happening. Cornetta's group also published their findings of no RCL events in 296 trial subjects. And to date, there still have been no reports of confirmed RCR or RCL reported in the literature. So the current status of RCL testing within our program, in terms of products, RCR/RCL testing is not a required test for release for most of our standard CAR T products manufactured in CVPF. The final product sample is archived indefinitely, but could qualify for disposal if a trial has been ended for more than 5 years or a patient has passed away, whichever comes latest. From a patient monitoring perspective, our post-infusion samples are collected and archived at pre-infusion baseline and months 3, 6, and 12 post-infusion. In the event an RCL is suspected, the archive product and the post-infusion samples are available for use in investigation. Switching over now to look at long-term follow-up. This is something that we are actively working on here. In our program, we have sort of 2 phases of follow-up for our patients. The primary follow-up is part of the therapeutic protocol. And here we're looking at the safety of a new CAR or cell product and its activity. So a lot of what we're focusing on in terms of safety is toxicity or off-target effects or dose relationship. Whereas in the second phase of the protocol, or the long-term follow-up, secondary follow-up, we're looking at integration, we're looking at the safety of the approach or the integration. So we are focusing on integration sites and concerns or implications, and what we call PDAEs, or protocol-defined adverse events. These include new malignancies, new incidence or exacerbation of a pre-existing neurologic disorder, new incidence or exacerbation of a prior rheumatologic or other autoimmune disorder, new incidence of hematologic disorder, or new incidence of infection, which could potentially be product-related. So the FDA put out a revised guidance again in January of 2020. This one is related to delayed adverse events. So it's a long-term follow-up after administration of human gene therapy products. And some of the key passages from this include, number 1, the agency considers the assessment of risk to be a continuous process. As more data accumulates, they recommend you reassess the risk to your subjects, and if appropriate, revise your existing long-term follow-up observation study protocol. Another key statement here is that the preclinical and clinical experience with your product or similar products may be considered relevant in the assessment of risk for delayed adverse events. For example, experience with gene therapy products in the same vector type administered by a similar route or given for the same clinical indication may contribute helpful information. And one of the big things that we highlight from this guidance here is to reduce the unnecessary burden to study subjects. And as a study sponsor, it may be appropriate to modify the duration of the long-term follow-up based on your ongoing assessment of product persistence, transgene expression, and clinical findings. So this was really the statement that opened the door for us from a long-term follow-up perspective as well. One concern the FDA has is the development of leukemias and pre-malignant conditions or clonal expansion due to lentiviral vector or gamma retroviral vector integration. They recommend analysis of vector integration sites if greater than or equal to 1% of the surrogate sample is positive for vector sequence by PCR. And in our publication that I've referenced, we reported an estimated median time for lentiviral modified T cell products to fall below that 1% threshold for 1.4 months in hematologic malignancies, 0.66 months in solid tumor, and 0.92 months in HIV, indicating such events would be captured well before the end of the current recommendation for long-term follow-up. So as I've alluded to, we are currently exploring compiling comprehensive safety and persistence data to support reducing long-term follow-up duration. And some of the things we've been able to do thus far, we've been able to close 2 INDs for HIV lentivirus modified cell therapies prior to 15 years based on lack of persistence and a favorable safety profile and lack of PDAE. We recently amended an adult ALL protocol, now performing only annual visits via phone or telemedicine after month 24 if CAR T cells are no longer detectable. This is a change from previously we were following patients in person out to 5 years. We've leveraged previous CAR-T data for a new study in breast cancer, where total length of follow-up is now only 5 years post-infusion. And we're currently amending an HIV CD4 CAR CCR5 zinc finger nuclease trial to conclude the follow-up after 5 years based on previous CD4 CAR trial data 15 years out, and 2 CCR5 zinc finger trials with data out 5 to 10 years. And as I've mentioned as well, we're currently working on a manuscript detailing our comprehensive programmatic data. So essentially the goal here is finding a balance. We are looking at long-term follow-up of 15 years versus a shorter follow-up duration. When we try to look at 15 years, we should ask ourselves what, obviously we have more data that's being generated, but is it really informative? Is there an active concern being addressed? And how much safety data is needed to inform on next generation products? On the flip side, looking at shorter follow-up, questions to ask would be, how long is long enough? Are there risk considerations that need to be taken into account if reducing the duration of follow-up? And really, what is the value of impact on costs, resources, and patient burden? And that's all I have, so thank you.
Thank you, Julie, for that initial discussion. Hi, everybody. My name is Jonathan Jazayeria. I'm an executive director at Kite Pharma. We're going to be discussing a case study today, discussing the data extrapolation for next generation cell therapy products. Today's case study includes 2 products. Primarily, I'll be talking about Tecartus, or brexacaptogene autoleucel, and its development and accelerated development following axicaptogene ciloleucel, or Yescarta. These are 2 distinct CD19-directed CAR-T therapies, and as I'll be discussing in a moment, the opportunities for data extrapolation and the success that we had with these programs. The first, I want to give a little bit of background in terms of the differences of these 2 programs. The first is that these are 2 distinct products. They do share an antigen that they target, and they also share the same vector that's used in manufacturing. However, there are notable changes in the manufacturing process, and these changes in the manufacturing process have resulted in a change in the therapeutic effect. Among the differences in the manufacturing process, first is that there's a difference in the apheresis material. One takes lymphocytes and the other, Tecartus, takes enriched T cells. Another notable change is a difference in the T cell activation step. The media does differ between these 2 products. And as I just alluded to, some of these changes in manufacturing also result in an observed enhanced effectiveness against circulating tumor cells. So there is a change in the therapeutic effect as a result of these manufacturing changes. Based on recent FDA guidance for industry and interpreting sameness for gene therapy products, this similarly follows that guidance in that the changes made to Tecartus resulting in a change in the therapeutic effect now results in a new product. Of note, Tecartus did not pursue the same indications as Yescarta, and I'll discuss in a moment in subsequent slides, and we'll touch on this a bit during the panel discussion, around differences in strategy for next generation products, whether those be to pursue the same indications or whether those products are pursuing a replacement strategy to pursue the same indications as the primary product. A little bit about the development history for Tecartus. In September 2015, there was a conversation between FDA and Kite, and there was an agreement to submit a new IND for what was referred to as the XLP process for axicell, which eventually became called Tecartus. In October, the Tecartus IND was submitted, and following that IND submission, the ZUMA-2 protocol, which was originally dosing patients with MCL with the Yescarta product, the axicell product, this was the first time that Tecartus, or the XLP process, was being used in a clinical trial. Following that, in October of 2017, Yescarta was approved by the FDA. And in April, the FDA did require a new BLA, a separate BLA with a separate brand name and a separate USPI for Tecartus. And finally, in July of 2020, Tecartus was approved by the FDA with an accelerated approval for MCL. Some examples of data extrapolation that were used for this particular example. And first, I'll start with just the starting dose. It's important to note here that the starting dose for Tecartus was based on the approved clinical dose for Yescarta. There were a few doses explored, so 2 dose cohorts, but there were no staggering or other type of typical first-time in human safety measures that would have delayed or postponed Tecartus's development in MCL. The other is the stability data. We do have results from stress studies that were used and extrapolated across to confirm the analytical methods, as well as the container closure integrity systems. These were used to support stability, but unfortunately, the long-term stability conclusions were not able to be extrapolated. And finally, the safety data. The FDA agreed to extrapolate safety data from Yescarta to Tecartus, and that resulted in a combined REMS program. Now, it's important that the starting dose, I'd like to highlight, this is an area that several programs have issue with the agency, because every time we develop these next generation assets, the approach that we have to start with is essentially starting from square one. But this is an example of where a next generation cell therapy product made it to the clinic and was able to accelerate or skip some of these traditional first time in human steps, and ultimately resulted in a successful program which was able to deliver a treatment to patients sooner. And finally, on my last slide, I'd like to summarize what we'll be discussing a bit further in the discussion today, as well as in the white paper, the notion that not all next generation cell therapy programs are the same. They may have different level of changes, which results in different opportunities for extrapolation, but also the intent behind what the program is looking to do may be different. So for instance, if I take you in the first category, changes in manufacturing, this is similar to the Tecartus or the Tecartus-Yescarta example. Here we have products with the same antigen, same vector, but there's a change in manufacturing. Now, the company could choose to pursue new indications, like the Tecartus example, or choose to pursue the same indications, or like a replacement strategy. And we've seen this in the small molecule and monoclonal antibody space, but we have yet to see this be successfully done in the cell therapy space. And this is one where an initial product, a primary product, is going through development, and a secondary product looks to leapfrog and essentially replace where the manufacturer has no intention of manufacturing the initial primary product any longer. This could happen at different stages of development, early, late, or even for a commercial product, where a next generation program is seeking to displace the existing product. This has the greatest opportunity for data extrapolation versus the middle column, which we'll discuss now, which are those programs where the new next generation asset seeks some enhancement that is also changing or improving the existing clinical therapeutic effect. So here's one where you have a same antigen, maybe a same vector, but the vector has some enhancements to it. The plasmid inside of the vector may encode for certain transgenes. These could be things that infect the tumor microenvironment or binding affinities, but essentially you're targeting the same antigen. You could pursue new indications or the same indications, like we just discussed, a replacement strategy. But here, your opportunity for data extrapolation is a little bit more moderate, because you don't have all of the same considerations in the first column, where you're just changing the antigen and you have the same antigen and the same vector, but maybe making some tweaks to the manufacturing process. Whereas here, you're now introducing some altered biological activity. Versus the third column, and this is maybe the one with the least amount of opportunity for data extrapolation, but one where you have a new target, a new or dual antigen. You're introducing a new mechanism of action, maybe having new tumor types or new histologies impacted. Presumably, the strategy here would be for new indications, where a replacement strategy is less reasonable and would be more challenging, I think, from a regulatory perspective. And as I mentioned, probably has the least opportunity for data extrapolation, but one where there may still be relevance that can carry through. So I want to thank you today for listening to me, and we'll have further discussion and extrapolation on these points in the white paper and in the panel discussion. I'd like to hand it over to Jeff for further comments.
Thanks very much, Julie and Jonathan, for those really insightful presentations. As a reminder, please feel free to write a question in the Q&A section for our panelists. I'd now like to turn things over to our panel today, which will be moderated by Michael Kalos from Next Pillar Consulting. Michael?
Well, Jeff, thank you for the kind introduction. It's my pleasure today to be joined by our four panelists, Mark Better from PharmEffects, Julie Jadlowsky from the University of Pennsylvania, Jonathan Jazayeria from Kite, Gilead Company, and Ingrid Markovic from US FDA. So let's kick this off with talking about our progress to date, or in other words, where we are today. The case studies that we heard today highlight the evolving landscape for generating data to support the development of cell-based therapies. Kicking it off, let's start talking about what key regulatory advancements we've seen in the space for the past 2 years that either have or are influencing the clinical development and strategies of programs currently under development. Ingrid, maybe you can kick it off with a perspective from FDA for us.
Sure. Well, thank you, Michael, for this kind introduction. It's a great pleasure to be here. And I think the regulatory advancements are most informed by scientific discovery, and FDA truly looks to the field to share information to inform our thinking about regulatory framework development. And just to share some of the recent opportunities, there is a CMC development and readiness pilot program, which is intended to facilitate CMC readiness for some of the highly innovative therapies, and most of them could be in cell and gene therapy space, with breakthrough or RMAT designation, so CMC can keep up the pace with clinical development. And for folks who are interested, there is a Federal Register notice that announced the pilot, and CBER will be receiving applications starting from April 1st. So everybody is more than welcome to check this out. And to this end, I think it's also helpful to reflect on some of the past examples of regulatory advancement, like breakthrough therapy designation, and really applaud the efforts of Friends of Cancer Research for spearheading this initiative, which was then signed into law about a decade ago, and then became an international effort with PRIME designation for EU and Sakigake in Japan. And just to highlight a few recent cell and gene therapy guidances that may be helpful advance the field, we have on top of the list, perhaps, is studying multiple versions of a cellular or gene therapy product, which is also referred to as primary secondary IND guidance, which provides pointers how to study multiple versions of a product under single clinical protocol, namely an umbrella protocol, in which case the clinical, preclinical, and CMC information could be captured in the primary IND, and additional CMC information and preclinical info for new product version could be provided in the secondary IND with necessary cross-referencing. And the idea behind this is really to help reduce administrative burden on both the sponsor and the FDA. There is also the sameness guidance that might be of interest, which allows exclusivity while promoting innovation in cases where there is a significant improvement in safety and efficacy. A couple other guidances that may be helpful to flag is the draft CAR-T guidance, the draft genome editing, and also not posted yet, but it's on the list for the 2023, is the comparability guidance, which is a very hot topic, and we had a lot of very helpful discussion. And I think it's also helpful to mention the omnibus bill, which could be very pertinent to this discussion. It's just hot off the press, passed in December of 2022, and there's still a lot of discussions in the FDA. What are the implications of this? But two provisions could be especially helpful here. The Section 2503 on platform technology, which allows sponsors to reference or rely on information in one application that uses platform technology to support the approval of a different application, and also advanced technology Section 3213 with a similar idea.
Thank you, Ingrid. That's terrific. Jonathan, some thoughts on from your perspective on key advancements over the last 2 years?
Sure, no, absolutely. I'd just like to echo a few of the points that Ingrid mentioned, specifically calling out a few guidances. The studying multiple versions of related products, I think, is one that does a good job of codifying a framework that's been in use by some sponsors, but I think it does share the learnings that the agency has had across those from those programs and those sponsors that have implemented such designs, especially with regards to master protocols and more unique and progressive trial designs. The other one I'd like to highlight that Ingrid had also mentioned was the sameness guidance, or I like to call it as the sameness guidance. Specifically, it was designed to talk about orphan drug exclusivity, but I think there are tenets within this guidance that serves well to indicate where the FDA's thinking on is on the age-old question of exactly how different is different, and when is it one product and when is it a new product. So I think some of the tenets and the comments in this guidance can be carried through to some of the items that we're working on and discussing through this panel in this white paper.
Terrific. Thank you, Jonathan. Continuing on the thread of progress to date and where we are, maybe we can touch a little bit on what's needed for us to continue to make scientific and regulatory advancements in the most effective manner. Julie, would you take the lead on that?
Sure. Thanks, Michael. So, first off, I want to say the FDA has been very accommodating in the past few years. I know we've all been through a lot with COVID. There's been a lot of turnover at the FDA that we completely appreciate. But I think maybe one of the biggest struggles that we've had is a little bit of a disconnect when it comes to reviewers. So, I come from the academic perspective at the University of Penn. We do a lot of phase 1 studies, a lot of small studies, but they're all very similar from a manufacturing perspective, from a preclinical perspective. And we find that when we are submitting these INDs, there's some discord between reviewers. And it may be that they're newer reviewers and they just don't have the historical perspective, or maybe there's not a lot of alignment or talking that goes on behind the scenes about some of these similar situations. So especially in things like potency or specificity or even release testing, when we're submitting these preclinical packages, we're getting a lot of divergent feedback, which is a little bit frustrating from our perspective, because at that point then we are looking at potentially a clinical hold because we haven't addressed a particular issue that was never an issue in the past. So I think, from our perspective, it would be great if we could have more conversation, maybe develop frameworks or best practices that would help address some of these things. But then, on our end, I think we need to also fulfill our obligations of getting our data out there into the public realm, because I think we learned from the RCL issue that the FDA is very open to changing things, but if there's nothing in the public domain for them to reference, it's really hard, because they know these things that are happening, but they're all specific, and they can't be sharing them across INDs to different sponsor groups. And so I think even when data is maybe not incredibly interesting or might seem trivial, it's important to get that negative data out there in publications so that we can spark some of this conversation and give the FDA a leg to stand on when it comes to making some of these advancements.
Excellent points. Thank you, Julie. Mark, anything from your perspective to add to this?
Yeah, thank you very much. Having watched this cell therapy field evolve over the past 10 years or so, when I took my first job in a sponsor company developing a CAR T-cell program, we've progressed from an environment where both sponsors and regulators were unclear on the path to product approval for engineered cell therapies, to an environment where a growing number of products have now reached the market. It's both product and process, as well as clinical understanding, that have allowed these advances, with more established protocols and procedures for advancing programs and products. Part of our overall understanding comes from testing hypotheses, of course, but certain standards are being set for both running cell therapy development programs, as well as pathways for regulatory approval. From my view, with more and more information available, the pace of advancement in cell therapy product development will certainly increase dramatically.
Thank you, Mark. Let's touch a little bit now about where we're going based upon what we've heard and other information, of course. And let's talk about a risk-based approach for product development. The question it poses is, how do we enable a more streamlined and efficient product development paradigm and enable the iterative product improvements by applying learnings across different generations of the products? Essentially, to design and execute the next generation of products in a more innovative and time-efficient manner. Ingrid, maybe you can take the lead on that and talk to us about how the FDA is thinking about this topic and issue.
Yes, of course. Thank you, Michael. So no two products or programs are exactly the same, as I'm sure you're aware. So identifying the commonalities, including shared learnings and differences across product generations, and start building the case by kind of understanding the impact of those differences on safety and efficacy or both would be a good starting point. And also having a better understanding, I think something that Jonathan mentioned, what constitutes a product version. And so there may be a very helpful examples in the appendix of the primary, secondary IND guidance, which could provide some general pointers, but it should not be taken as an all-inclusive list. Another thing that may be helpful to be mindful of is really separating the routine manufacturing changes for the sake of process improvement and optimization, which are generally not intended to generate different product version. These routine manufacturing changes are generally subject to comparability assessment, whereas generation of a different product would not be, because we would anticipate to see minor product differences in the latter case. And one other thought that may be of interest is in sort of for the sake of shortening the development cycle, sort of thinking along the lines of how do we streamline, sometimes it may also help to take a step back and look at holistically at all the development operations and identify any weak links. For example, there may be a raw material supply chain vulnerabilities. Something could be happening on the manufacturing floor, or it could be at a patient enrollment site. So there is a whole myriad of things that could potentially cause unplanned interruptions in operations. So anticipating these ahead of time could help coming up with a mitigation plan, and it could really go a long way to inform and streamline future development.
Thank you, Olga. Jonathan, maybe you can speak a little about the important elements and considerations for developing a risk-based framework.
Yeah, absolutely. Absolutely. Thank you. So again, similarly, piggybacking off several of the points that Ingrid was just mentioning, not all programs come to the clinic, come to the table, the same level of risk or unknowns. There are multiple ways that you can have product variations. And the primary product that we're discussing and the one that you're trying to extrapolate data from could be a program that's similarly in early stage development and more of a mid or late stage development, or could be an approved product. And the amount of learnings and data that's available on each of these programs as it goes through its product lifecycle will increase the opportunities for that data extrapolation. I think the agency could also consider developing or discussing pathways for new products to be developed, not only with superior clinical profiles, but perhaps comparable clinical profiles. We have examples of this with more traditional molecules like small molecules, monoclonal antibodies, which do provide better opportunities for patient use. They may be things that increase half-life or those types of developments. In the cell therapy space, we could consider manufacturing platforms or improvements in turnaround time in a similar vein, where it can improve patient access, but not necessarily have to go through the same clinical rigor as a novel program trying to seek a superiority claim and appear on the market in that fashion.
Fantastic. Excellent points, Jonathan. Thank you. Mark, perhaps you can speak to us about what foundational and key information we might need to know about in terms of the relationship between a primary and secondary product that would allow us to support the type of data extrapolation that we're discussing today.
Yeah, thanks, Michael. As we think prospectively of cell therapy development, where iterative versions of a product may be introduced over time as our overall product knowledge increases, it's extremely important to have robust tools to understand what the product's biological properties really are, and to be able to use those tools to guide development. Analytical methods used for product release and importantly for extended characterization are essential to establish relationships between primary and secondary products. As we learn more and more about what our analytical tools tell us about a set of similar products, we'll be in a stronger position to support a risk-based product evolution. Perhaps most importantly are those analytical tools that can assess biological function, or what we commonly think of as our potency methods. It's in a sponsor's best interest to implement well-characterized and qualified potency methods as early in development cycle as possible to increase product knowledge, and where feasible, to implement a set of orthogonal potency methods to maximize product understanding. For example, with engineered T-cell products, it's often useful to establish measures of target cell killing or inhibition, as well as methods to measure cytokine production after incubation of the cell therapy product with antigen-positive target cells. Methods to detect expression of multiple transgenes or detect gene knockout also set the stage for understanding how the function of a primary product might relate to the function of a secondary product. Often these characteristics that link a primary product and a secondary product are evaluated in non-clinical models, whether they're in vitro models of activity or in vivo models in animal studies. These tools are very useful often to guide us as we think about what product characteristics we should look at for part of character or releasing characterization over time to make sure that our processes remain under control. And lastly, in addition to the CMC and the non-clinical activities, there's potential for clues to evolve from clinical studies that might help guide the evolution from a primary product into a secondary product. For example, are there safety signals in initial trial that can be addressed by an improvement to the cell therapy product? Or does a clinical safety and efficacy profile suggest an opportunity to further improve outcome that might lead to a secondary product? I think at the end of the day, we need to take a holistic approach to product development and extrapolate data where possible from non-clinical CMC and even clinical learnings to guide product development, and maximize the potential to benefit patients in human studies.
Thank you, Mark. Excellent points in summary, I might add. Ingrid, perhaps you can speak to us about what are some of the important considerations for a developer to consider, understand, and address as we assess or as they assess product characteristics and product quality.
Yes, thank you, Michael. So echoing some of the really good points made by Mark and Jonathan, also understanding the key attributes that have a clinical benefit that we want to study in a clinical trial, and the ways to tease them out during manufacturing could really go a long way. For example, if you have a situation where the starting material is highly variable and could contribute greatly to product variability, having a well-designed manufacturing strategy could effectively reduce this variability and result in a relatively consistent product. And related to this, also identifying the relevant analytical methods that measure these attributes is important, as Mark also mentioned, and also understand where existing methods could be used to, could be leveraged to support the next generation product. As we know, method development and qualification is very time and resource consuming, so understanding what methods have broader applicability could help save time and resources. And we also need to keep in mind the life cycle of the product, where our knowledge of product quality attributes, how to measure them, understanding of the risks may actually change over time as body of knowledge about the product and process, as well as clinical experience increases. So our work really is not done even when product is on the market. We continue to assess the risks and evaluate what works best for the patient.
Thank you, Ingrid. Excellent points. Maybe we can touch a little bit on variability in data. We're talking about a lot of data today and leveraging data to inform us to do trials in a more effective and efficient way. Mark, how would you propose developers deal with the variability in quality, variability in benchmarking data coming from different sources? And how does this impact our ability to leverage the data? For example, data coming from different manufacturing sites, different clinical sites, data coming from academia and industry. How does all of that, how can we look at all of that in a holistic manner?
Yeah, I think it's important. The variety of sources of product and process knowledge that can be leveraged when we're thinking about, development of a specific product, primary product. And often additional information then comes into play when we're talking about developing a secondary product that adds on the information that's available from the first product. So when thinking of a risk-based approach to product development and opportunities for data extrapolation, it's certainly appropriate to look beyond the data set that might be available from in-house or sponsored work, notwithstanding the complexity that evaluating data from multiple sources might bring about. When we think about risk-based product development, any known or suspected variability in starting materials, Ingrid mentioned, analytical tools, clinical trial design, et cetera, I mean, definitely need to be taken into consideration carefully. Since cell therapy is still a relatively new field compared to other therapeutic modalities, the industry hasn't yet coalesced on standard tools and strategies, which adds to the complexity that we have on us as developers. Differences from sponsor to sponsor, or lab to lab, they're fine, but any differences need to be carefully considered and understood as part of an overall risk program for the product development. Like all product knowledge being considered for risk-based development, literature data from similar products developed elsewhere by other sponsors, data in publicly available repositories, as well as any sponsor data need to be considered carefully and in combination to look for gaps and similarities so that we can develop the products as efficiently as possible, but understand where these gaps might be really critical for revealing new avenues for investigation. So it's quite complex to look at these different sources of data coming in, but also it's very important. And overall, having a larger database of product knowledge is always useful, even if there are some gaps that can be evaluated.
Thank you, Mark. Let's pivot now and talk a little bit about regulatory policy and how do we get to where we want to be. Specifically, how should we think about data extrapolation in the context of bringing data together for an IND submission, for an IRB submission? Julie, perhaps you can take the lead on that question.
Sure, I can talk a little bit about an IRB submission. I think in terms of an IRB submission, we're really focusing on the protocol and on safety. And so the protocol is a good place to take a look at your previous data and see what you can extrapolate in terms of toxicity management or potential adverse events, what might be expected. Also thinking about how to use that data. So you can use that data potentially to help guide what your starting dose should be, or whether you should use a fractionated dosing strategy in your protocol, or even using the established MTD, or taking some of that data and trying to use it to justify doing a retreatment strategy in your patients. I think the more data you get, the more you can use to kind of push the envelope a little bit. And I think especially in our program, we're very much doing the same manufacturing, as I mentioned, the same sort of approach to everything. And so using the same vector backbones and using the same manufacturing strategy gives us an ability to kind of take that programmatic look at it and try to apply that to the safety, to try to make better protocols, try to get things moving a little bit faster.
Thank you. Jonathan, any thoughts on your side?
Yeah, when I think about this problem, I think about what are we trying to solve for? And I think what the issue that impacts, I think, all sponsors is we have these next-gen programs that seem to be more promising. We take them to the agency, and the feedback we tend to receive is that we're kind of starting from square one. It's a new product, unknown entity, and therefore we have a lot of similar type of feedback in terms of dose starting, dose range finding, DLT definitions, and things of that nature. As Julie mentioned, I think there may be opportunities to leverage this data holistically across. I think new IND submissions should be encouraged to include, perhaps in Module 2, sections that specifically provides context related to what data is being extrapolated from which primary products and for what purpose. For instance, are we trying to use that data to negotiate or to discuss why DLT definitions should be different, a starting dose, discussing staggering of patients between cohorts or within cohorts. All of these things are issues that I think that impacts several sponsors, if not all sponsors in this space, when we're trying to develop these next-gen assets.
Thank you, Jonathan. Jonathan, can you speak about key learnings that can be extracted from your interactions with FDA regarding Yescarta and Tecartus?
Yeah, no, absolutely. Thank you for bringing that up. So as many folks know, there are two programs, two approved products. They are both targeting CD19. There are a number of similarities between the programs, but they are distinct assets and distinct products. However, during the development of Tecartus, which was the secondary product, if we use the primary and secondary terminology, which didn't quite exist at the time, Tecartus was able to leverage the data that was available with Yescarta in MCL, for instance, which was the lead indication for Tecartus, and was able to extrapolate some of the data. They didn't have to go through robust dose range finding. There were a couple of doses that were explored, but we didn't have to do the same sort of dose staggering for a first time in human study that one would have to expect or be experienced today. And so we have seen successes with that type of extrapolation approach. We've also used similar extrapolation approaches between those two products from a REMS perspective, where both products share a safety REMS program. There are other ways that we were able to extrapolate some of the stability data, for instance, and confirm analytical methods and certain testings to support our stability programs. Long-term stability wasn't carried through, but I think if you don't build the avenue, cars aren't going to drive down it. So I think we need to discuss a framework that will allow more sponsors to share and discuss with FDA opportunities for data extrapolation so that we can do this for more programs.
Thank you, Jonathan. From a regulatory framework, let's talk a little bit about what we might need to include to be able to apply the learnings from first generation products in the rapid development of the next generation products. Jonathan, maybe we can continue with you.
Sure, absolutely. Just sort of continuing from what we were just discussing, I think the FDA should formally encourage sponsors to introduce and discuss their extrapolation strategies, perhaps even preparing or developing some sort of a document similar to an assessment aid, something that sponsors can use, that they can explain their extrapolation approaches and assess these extrapolation approaches as being whether a high risk, a medium risk, or a low risk, and why they believe so. And I think once the agency gets more of this exposure and experience from different sponsors, they can begin to build that framework and have that comfort for when to allow for certain extrapolation and when not to. It may start off fairly conservative, but again, like I mentioned, if you don't build a path for having these conversations, then we'll never be able to do so. And I think this could be the first step in having those engagements.
Thank you. Ingrid, your thoughts?
So I think this is really great feedback, and thank you so much for sharing. I'm more than happy to take this back. But I think it's also important to briefly reflect on how far the field has gotten overall in teasing apart different aspects in the design space, which I think is a huge accomplishment. And I think it would be helpful to recognize everyone working in this field. And certainly, I think this is a great opportunity, just what we have right now. And FDA is certainly willing to continue having an open dialogue, as we kind of all as a field learn how, what works best for this space, to help formulate the best regulatory pathway to fit the product, as well as a clinical development strategy. And as we continue to inform the field through active listening and communication and outreach, I think it's also important to reiterate that we certainly take industry feedback very seriously. And, in a number of formal or informal ways, we're happy to communicate our current thinking, some of which has been through guidances. And there are more interactive opportunities like town halls, public workshops, where we have an open dialogue, and folks can certainly feel free to bring their thoughts at that time. And I think opportunities for partnership to exchange ideas in general are very helpful to advance the field and make sure that these transformative therapies get to the patient as fast as possible.
Thank you, Ingrid. Julie, any thoughts from you on this topic?
No, I mean, I agree with everything that's been said. I think, in general, I think we've been making a lot of improvements, a lot of advances in a short amount of time. And I think this is a great forum to do that. And we should definitely keep it up and keep pushing.
Mark.
I'd like to say that I'm very optimistic about the field these days, because I think sponsors are becoming much more informed and focusing more on the critical aspects of product development. And what I see from the regulatory agency also is that there's more and more guidances coming out, more advice. I know everybody's really busy, but nonetheless, I think the total amount of information is increasing dramatically, and that's only good for product development. So,
thank you. Thank you all for your comments, input, and thoughtful discussion. With that, we'll turn it back to Jeff.
Thank you for joining us today for a robust conversation on cell and gene therapy. A special thank you to everyone who participated today and to our incredible working group who made this possible. Following this virtual meeting, we'll bring stakeholders together for a hybrid meeting in Washington, D.C. on May 22nd to further discuss implementation of these strategies in a streamlined regulatory framework. Registration is now open for that event. You can click the link in the chat to register or scan the QR code on the screen. Additional information will be provided on our website as it becomes available, and a link to register can also be found there. In advance of the May 22nd meeting, we will be distributing a white paper that will provide a deeper dive into many of the topics discussed today as we use the May meeting to identify opportunities for implementing these ideas. We hope that you can join us. Thank you and take care.